All right, so in this particular lecture, we're going to talk about activated sludge. So this is where biological principles are going to start to be really, really important. So if there's any biology as we go through these types of treatment that's a little bit confusing, please don't hesitate to let me know. I'm going to try and keep it very high level when we talk about it in class. So activated sludge, it's a type of primary and sometimes secondary wastewater treatment. So it comes after preliminary, but before kind of those polishing steps of tertiary treatment and disinfection. So what it does is it uses biologically active components of influent wastewater to digest organics via cellular respirations. So basically what that means is that there are microorganisms in wastewater, and they use organic material as their substrate, which is basically their food. And then under aerobic conditions, meaning that they have oxygen in the system, they're going to use that substrate to perform what we call aerobic respiration. And so basically what that just means is that the organisms are using oxygen and they're feasting on all of the organics that are in the system. So they're eating what's there and they're using oxygen to do that via cellular respiration. So the purpose of activated sludge treatment is specifically to break down those organics and to consume organics in wastewater. It's also to help us settle out some of those settleable solids from activated sludge process. So there's still going to be solids that can settle out of the system. And then one thing that we also have to do is we have to think about maintaining oxygen in the system using aeration. So remember, when we have high BOD, it pulls lots of oxygen out of the system. And so if organisms are very active in the system, there's going to be higher BOD. So we add oxygen to the system many times in order to make sure that those levels stay relatively stable. So forgive the kind of funny diagram, but how does activated sludge work? So in our influent wastewater, we have BOD, organics, solids, and then we have microorganisms. So this is our influent wastewater on the left side. And there's two steps in activated sludge most of the time. The first step is what we call an aerated basin. So in this basin, you're going to have the BOD, the organics, the solids, the organisms, and we're going to pump in air in the bottom. We're going to have mixing. So this is meant to be a little paddle for mixing, like a flocculator. And we're pumping in air, so all of these little white bubbles here represent oxygen. And so the combination of these organic solids and microorganisms, we call this mixed liquor suspended solids. And so the idea here is that we're getting everybody together, we're pumping oxygen in the system because organisms can use that oxygen and can use the organics to do cellular respiration. So that's the first step. The second step is to pump what comes out of the aeration basin into what we call a secondary settling basin. So this secondary settling basin, at this point, the organism has enough oxygen to basically break down those organics. And as it breaks down those organics, it grows and it divides and it makes more microorganism, it reproduces basically. And so what we get out of that combination in red down here is what we call activated sludge. Now, solids are also going to settle out of this process at the same time because it is a settling tank, but really what we're trying to produce here is this activated sludge. So it's a biologically active component. It's constructed of solids and then broken down organics. So hopefully most of the organics have kind of gone away because the organisms have been eating them. So the activated sludge contains those microorganisms and some of those solids, and it's basically this big thick mass that has lots of activated microorganisms in it. And so what we do with that mass is we recycle it and we pump it back into the aeration basin to put it back into that mixed liquor, suspended solids. And basically what that does is that keeps the process going over time. So microorganisms are going to have to split and divide and reproduce in order for this to be a viable type of treatment. If you kill off all your organisms, this won't work. So the purpose of recycling that is to keep that activation of microorganisms. So basically what's happened is within these two steps, microorganisms have consumed organics, and they're now sitting in the activated sludge with pretty much no organics to deal with, right? Because they've consumed them all. So if we recycle them and we bring them back to the aerated component, there's going to be organics in this aerated component again, and that activated sludge is going to interact with those organics and it's going to keep that process going over time. So that's why we recycle that sludge into the system. We also waste a certain amount at some point. That is not going to be a viable solution based on how many cells are in kind of that activated sludge from a microbiological standpoint. So that's kind of the idea. We have influent wastewater coming in, maybe from a primary treatment, from a preliminary treatment, depending on the system. We have an aeration basin where we're having contact time in the water. We're mixing everything up and we're putting more oxygen back in. The microorganisms are consuming those organics in the presence of oxygen, and that activated solids and microorganisms is your activated sludge that gets recycled so that those organisms can continue to break down organics in the system. And that's the idea behind this. So this is a little bit more of a simplified diagram of the exact same thing that we just showed, right? So you have influent water coming in, you have activated sludge being recycled to that aeration basin, that secondary clarifier pushes the product water out. And so what you can see on the bottom are two different pictures of what this actually looks like. So the fact that you can kind of see this foam on the top of this, that's because of the aeration. Those are like bubbles coming up from air. So that's actually what you're seeing right there. And these structures on the left, these are aeration pumps to keep air being pumped through the system. So key concepts here that we need to be aware of. Activated sludge uses an organism's natural tendency to break down organics to our advantage, right? So this is what they do naturally. That's what we want them to do in the system. We want to keep our oxygen levels stable with aeration. This is going to be really important because if you have high BOD content, we're going to consume lots of oxygen in the water. And we want to have enough oxygen that the organisms can still perform cellular respiration. And then the other thing is we want to keep our organisms well fed. So they need sufficient substrate. So they need enough organics. Otherwise they die off. And that's really not what we want. If we kill the microorganisms in our system, we no longer have organic removal. So we have to keep the organisms happy by loading pretty regularly with a consistent mass loading of organics. The other thing that's really important is to have that recycle, right? To make sure that we're continuously pulling organics out and then pushing the organisms back in to interact with new organics in the system. So those are some things that are really important about activated sludge. So some components, right? You have an aeration tank, a secondary clarifier, you have a recycle line, and a waste activated sludge. And so that's really important. The fact that we have recycle lines, the fact that we're wasting things, the fact that there's two steps in this activated sludge is really important. And the other thing that I'll point out is this equation, generalized equation at the top of the slide. Basically, this is what's happening in the activated sludge process. We're taking organic matter with oxygen, and then aerobic microorganisms that, like oxygen, are going to produce new cells, new energy for their cells, CO2, water, and other end products. So we get rid of that organic material, and we're just producing water and carbon dioxide, hopefully. So some key parameters that you're going to need to know that we're going to discuss in class. You need to know what's called the food to microorganism ratio, or the FM ratio. And basically what this means is that if we have too much food or too much substrate in the system, the organisms can't break it down. You've basically given them too much food and they don't know what to do with it. And what will happen is the organics will break through the system. And that's not a good thing to do because we don't want organics leaving the activated sludge process. So if you have too much food, you're going to have organic breakthrough. If your microorganisms, if you have too many of those and not enough food, you're actually starving your microorganisms and they will start to die off and you will lose efficacy in your activated sludge process. And they're going to start to like outcompete each other and they're just going to die off. And so it's going to be less effective as a treatment. So this ratio is really important from a design perspective. We also need to know biochemical kinetics, so we need to know how organisms break down organics via cellular respiration. So we talked about chemical kinetics a lot in class. We also now need to start thinking about how do we do kinetics for biological systems, and we'll talk through that. You need to know your influent TSS and your BOD in order to understand what's coming into your system. And then you need to know the amount of oxygen that's going to be added in aeration because that's a really important step in this particular process. Some key calculations that you need to know that we're going to talk about. We need to know the recycle rate. we need to know how much sludge is going back into the aeration component. If you push too much in, it's going to consume all those organics and we're not going to have enough contact time. The oxygen rates are going to go down. We need to be aware of that. We also have a parameter called mean cell residence time. So that basically is a way of looking at how long do cells typically stay in each basin. It's kind of similar to a detention time, but we're focused on the biological component. Mass balances are actually pretty key with a lot of these systems. We need to know how much sludge is moving through the system. And then the FM ratio is really important because it tells us how much of the substrate is being utilized. So last couple things, some advantages, very little addition of chemicals in this process. That's really good. And it recycles sludge by design, so that improves overall performance and it cuts down on wasted material. So that's really kind of a nice benefit of this particular treatment. Some disadvantages are that it requires monitoring of biological parameters, and that's difficult to do what we call in situ. So it's really hard to monitor on a daily basis with like a probe or something like that we'd put in the water. You actually have to plate microorganisms out, and that's very involved. So monitoring these systems is something that's kind of difficult sometimes. And then aeration can be costly depending on the system size, and it's going to require constant power. So that's really important to be aware of because we want that oxygen in the system. So if we're in an area where maybe power is an issue, that might be something to think about. And then the last thing to talk about is some potential impacts. So your wasted activated sludge, that needs to be treated, right? Sometimes that can go to the landfill depending on what's in it, but it does have biological components and those need to be properly handled. So that could be something to be aware of in this particular system in terms of a waste component. If your biological components in the system are not well maintained, the system may need to be restarted. So it takes time to kind of get that cell concentration, that cell mass, in order for you to have an activated sludge process that's stable. So we don't want to kill our microorganisms in our system because it takes a long time sometimes to get that stable because it is a living system and that's important. And then that presence of a biological component can lead to more operational and maintenance costs over time because we are worried a little bit more about keeping these things alive. And so we're constantly monitoring things like temperature in the system in order to make sure that they're alive, and that might just be a little bit more labor cost, a little bit more in terms of monitoring equipment that you need to buy. So that's important as well. So in class, we're going to talk a lot about how we balance microorganisms in the system and how to calculate all of those different parameters that we talked about.